Hi everyone, we are going to go over DNA and how we get from DNA to a protein. Here are our learning objectives for today. Okay, so first, you're going to answer in iClicker what you know about DNA. And then what you would like to know about DNA. Okay. So first, let's go over some important terms. So we have a gene. A gene is a sequence of nucleotides that eventually encodes a product, and that product is typically a protein. A nucleotide, remember, is this right here, where we have a sugar, a phosphate, and a nitrogenous base. So sugar, phosphate, nitrogenous base. A chromosome is a whole chunk of DNA that has lots of genes, right? So that's way down here on our little scale. Okay. A genome is all the genetic material in an organism, so that's lots of chromosomes. And so we talked about the nucleotide building block. So many nucleotides makes up a gene, many genes makes up a chromosome, and many chromosomes makes up a genome. So nucleotides have a particular way of pairing. So we refer to one nucleotide code letter as a base, and then a complementary pair, we call that a base pair. So we have adenine and thymine that pair together, and we have cytosine and guanine that pair together. So A by itself is a base. A paired with T is a base pair. So go ahead and answer this in your iClicker. How do nucleotides correctly pair in DNA? Okay. Now, when you are entering DNA sequences, just enter letters and don't separate the letters with anything. Just a row of letters is fine. So first we are going to do a practice where we are going to write the complement of a DNA strand. This is our DNA strand, okay? And we want to pair these bases to make a complementary base pair. So we know that A binds with T and C binds with G. So we would have A, T, C, G, and then so on and so forth. And you can finish that sequence in iClicker. Okay, and while we're thinking about this, each of these letters is the nitrogenous base, right? So the green circle equals, I'll just color that in for clarity, is the nitrogenous base. Okay, along the back here, we also have our phosphates and our sugars. So we have our phosphates and our sugars all along here. Okay, and these eventually form this long backbone. Okay, so we have a phosphate sugar backbone, and that supports our complementary base pairs. And it's along this side too, right? So this is how we end up with our double helix shape, is the combination of base pairs and our phosphate sugar backbones. So what are we using our DNA for? So our DNA lives in the nucleus in eukaryotes. And we want to get from DNA in the nucleus to proteins that are doing jobs, right? Proteins do jobs. Okay, so let's think about this as a cookbook. Okay, our cookbook is like our chromosome. Okay, it has lots of recipes or lots of genes. So one recipe in the cookbook would equal one gene. Okay, now if we were to make a copy or print out the recipe, this is a transcript of our gene. So the copy is like our mRNA complement. Okay, and then when we go from our recipe to our actual, delicious brownies, that's when we go from RNA to a protein. We're taking the information from the mRNA and we're making it into a product. Okay? So, transcription, this is where we take the information from DNA, info from DNA, and we turn it into mRNA. Okay? This is transcription. So we start with our DNA, and our RNA is going to be built based on the DNA code. The differences that we have between these two, they're both types of nucleic acids. Nucleic acids. The difference is going to be this right here. So with ribose, we have an oxygen right there. And with deoxyribose, we do not have an oxygen. Okay? So we do have another little difference, which is the nucleotide bases that we can have. Both DNA and RNA can have cytosine and guanine. RNA, though, replaces thymine with uracil. So in an RNA strands, there will be no T, right? There's no T. We have U's instead of T's. Another difference is that our DNA is double-stranded. Remember, we have our complementary base pairs and our phosphate sugar backbone. Complementary base pair, phosphate sugar backbone, all the way down. Okay, our RNA, typically in humans, is going to be single-stranded. So we're copying one side of our DNA, or one side of the DNA. Okay, so we are making one strand. Okay, so the pairing, by the way, for RNA is going to be AU, CG. So A is still going to be a thing. U is the only one that we're going to see that's different. DNA has AT and CG. RNA has AU and CG. So when we make DNA, we have an enzyme called DNA polymerase. Now, DNA is the thing that we're making. ACE, the ending ASE, equals enzyme. And an enzyme is a protein with a job, right? And this protein's job is to make long polymers of DNA. So DNA polymerase's job is to add nucleotides to make DNA polymers, right? So a long strand of DNA is a polymer. That's all that means. It's a long strand. So if DNA, okay, so this question in iClicker, the enzyme that makes DNA is DNA polymerase. So the enzyme that makes RNA would be what? Makes RNA, and it's an enzyme, so it ends in ase. It's got a job, and the job is making polymers of RNA. Okay, so RNA polymer ase, right? It makes RNA, making long strands is an enzyme, RNA polymerase. Okay, so how do nucleotides pair correctly in RNA? So go ahead and select your correct answer here. And answer this in iClicker. iClicker. Anytime we see a star up here, we know that this is an iClicker question. Okay. So now, if we have this DNA strand, we are going to transcribe the RNA strand that is complementary here. Complementary. Okay, now RNA pairs A with U, C with G. So here we have a T. Now this T is still going to pair with an A, right? A still exists in RNA, but this A, it can't pair with a T. It pairs with a U, okay? And then we have C and G, and then go on and finish the rest of that RNA sequence, please. Okay, finish that in iClicker. Okay, so in transcription, we are reading one side of the DNA, right? We're looking at the gene on one side. So our RNA polymerase is going to bind to a section of the gene called the promoter. And it's going to go from that promoter, it's going to move forward and pair RNA nucleotides with our DNA nucleotides. And we're going to get this chain of mRNA. And that mRNA is then going to go somewhere else. Okay? Don't worry about PCR right now. Okay? So, but remember that RNA polymerase needs a promoter. Okay, eukaryotes, we end up doing some RNA processing. This can also be called splicing. Okay, so what happens when we process mRNA is we are taking out some of the mRNA and leaving other parts. So these two sections are called introns and exons. Okay, now introns are in the way, right? They are in the way. So the introns are going to get taken out. The exons get expressed. Okay, so those exons stay, right? So there at the end, we have exon A, exon B, and exon C stuck together. Our introns are gone. Now this mRNA is ready to become a protein. It's not going to become a protein, but the information on the mRNA is going to become a protein. So translation is the process of making an amino acid chain from mRNA. And we can think about this like translating a language. There are two languages here. The language of mRNA is nucleotides. The language of proteins is amino acids. So we have to translate mRNA from the language of amino acids to the language, from the language of nucleotides to the language of amino acids. So we have our mRNA here. And now mRNA has to code up to 20-ish, 21 amino acids, right? So we have these sections of nucleotides called codons. A codon is a sequence of 3 nucleotides that encodes an amino acid. So this nucleotide here is going to give us a lysine amino acid. And then we have another set of 3 and another set of 3. And each of these sets of 3 gives us a different amino acid. Those amino acids get bonded together at the ribosome. They get bonded together at the ribosome into a polypeptide. So a peptide bond is what links these together. Remember, poly means many. So a polypeptide means many peptide bonds. So a long chain of bonded amino acids. Okay. So translation is going to involve 2 particular things. So we need to have a ribosome, and the ribosome makes the bonds. And then we have to have our translator, just tRNA. And that sort of reads the RNA, the mRNA. Okay. So here we have our ribosome. Our ribosome comes and sticks onto our mRNA, and it starts pausing at these codons. And at each codon, we have a tRNA come in, and the tRNA has a section on it called an anticodon. And this anticodon is the complement to the codon. So we got the codon here, and then we have the anticodon on our tRNA, anticodon. Okay, and then on the other end of our tRNA, we have the amino acid. So the amino acid that's coded here is methionine. So our tRNA comes in, the anticodon binds to the RNA, and our amino acid is delivered. Okay. And we have tRNA that pair with all of these amino acids. Okay. So now we're going to do some translation exercises. There is a better quality codon chart on Blackboard if you have trouble with this one. When you are writing these down, please use the 3-letter code. And if you're entering more than one amino acid, just separate them with a space. Okay, so when you are responding in iClicker, use the 3-letter code and separate amino acids with a single space. So here's our first one. We're going to encode the first amino acid in this little sequence of RNA. Okay, so I'm going to highlight this. We are going to do this one right now. Okay, so the way that we read this is we go in order from left to right. So we start with our G. G is the first nucleotide, so we go to the first column and we find G. G is here. So we know that we're working in this row. Okay. Our second nucleotide is C, so we go to the C column. It's right here. So we know that we're working in this column. Right? So now we know that we need to be in this square right here. Our last nucleotide is C, so C is in the second row here. So there is our codon, GCC, and it codes for alanine, ALA. Now we're going to do our second one. Our second one I'm going to do in blue. This is our second codon. We start with A, our first nucleotide. That's this one. So we know we're going to be in this row. Then we have G. We go over to the G column. We're going to be in this column. Okay, so we know we're in this square right here. Then we have C as our third amino acid, third nucleotide, and that's our second row. So that gives us AGC, our codon, and this amino acid, serine. Okay. So in iClicker, you'll have a question for the first amino acid. That's this one. And then you'll have a question for the second amino acid. That's this one. Okay. So yeah, so that's there. That's iClicker. All right, so now we are going to go from DNA to protein. Okay. So we're going to go first from DNA to RNA, which is the process of transcription. Okay. So oh dear, what have I done? There we go. Okay. So I'm going to erase this so that I can write right under it. So take a moment, write out what you think this sequence of RNA will be, and I am going to work alongside you here. So we've got our C. C pairs with G. G pairs with C. Then we have A. Remember, this is our special pairing in RNA. Okay, and then next we have T, 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 so that gives us A. Then we have G, G, A, that gives us C, C, U, that's another one of our special RNA base pairs. And then we have G, A, U. Okay, so that's a little messy. Let's put it maybe up here in a little bit neater format. G, C, U, A, C, C, U, G, A, U. Okay, I'm going to help with the first one, but then I want you guys to do the rest of the amino acids on your own. Okay, so I'm going to start with green. We're going to work on this codon right here. So the first answer in iClicker is the RNA sequence. Now we are working on the protein sequence. So this is the next iClicker question. So green, our first nucleotide is G. So we go down to the G row. We know that we are going to be working in this row. Okay. Our second nucleotide is C. Here it is. So we're going to be working in this column. And then our third nucleotide is U. That's right here, GCU. So our first amino acid is alanine. Okay, and remember, you're just going to write the 3-letter code. And separate with a space. Okay, so go ahead and finish the rest of that amino acid chain. There will be 3 more amino acids. And enter that into iClicker. And lastly, what did you learn about DNA? Let me know. That'll be it.